everyone, this is Dr. CJ and today we will discuss about the pharmacokinetic interaction and pharmacodynamic interaction in the topic of drug interaction in the subject of pharmacy practice BP 703 TT. So, we need to consider about the mechanism of the drug interaction. Mechanism of drug interaction we discussed in the last lecture also about the drug interaction and beneficial interaction and adverse interaction. Here, if we consider about the mechanism of drug interaction, then there are two, in vitro and in vivo. Here in vitro we studied and we discussed about the physical interaction and chemical interaction. Over here in this lecture we will discuss about the pharmacokinetic interaction and pharmacodynamic interaction. So first, pharmacokinetic interaction. In this category, the change in the response to the first drug is the result of an alteration in the concentration of the drug at the receptor site and is produce difference in a drug absorption, distribution, metabolism and excretion. मतलब यहाँ पे क्या होगा कि कोई एक ड्रग हमने ली है, if we have prescribed one medication to particular patient, then that will be altered due to the effect or due to the concentration of another drug at the receptor site, and due to this condition it will make difference or it will produce difference in the absorption, metabolism, distribution and in case of excretion. So this is the pharmacokinetic interaction. So first we will discuss about the drug interaction during absorption. Absorption of drug from the GI tract is modified whenever any drug interaction alters the function of the GI tract. If the function of GI tract that is altered, then the physicochemical characteristic of the content of the tract which affect the oral dosage form, especially if we are taking the oral dosage form, then physicochemical characteristic that is need to consider, then condition of the mucosa, then active and passive transport mechanism. मतलब यहाँ पे हमें transport mechanism भी consider करना है that move the drugs from the tract through its lining of the body fluids. मतलब active and passive transport mechanism of the drug, then interaction during the dermatomucosal absorption. Dermatomucosal absorption भी यहाँ पे हमें consider करना पड़ेगा. So these are the points that need to consider for the drug interaction during the absorption. We will see this, we will discuss this one by one. So first, alteration of GI tract, alteration of gastrointestinal tract functions. So here, if we are considering gastrointestinal tract and if we are taking two or more drugs, see in case of drug interaction, here we are taking two or more drugs. So there will be the interaction in the GI tract, and due to that the absorption it will be decreases. Absorption of any particular drug that will be decreased in the body, and this will reduce or prevent any pharmacological effect of drug. So over here, when there is the alteration of GI tract function, then such a things, or we can say such a things will be happen in the absorption of particular drug. So first we will need to consider about the motility. Motility, emptying time of the stomach, it varies with the intensity of the gastric motility. देखिए यहाँ पे अगर gastric motility किसी की भी change होगी, likewise the emptying time of the stomach that will be varies, that will be changed. Faster a drug passes through the stomach and intestine, smaller is the amount of drug absorbed. देखिए अगर कोई मेडिसिन जल्दी से यहाँ से पास हो रही है, जल्दी से स्टमक में से या फिर इंटेस्टाइन में से पास हो रही है, या फिर अगर उसका पासिंग एम्प्टिंग टाइम, एम्प्टिंग रेट वो बहुत ज्यादा है, then in such a condition the smaller will be the absorption of drug, or we can say the drug absorption that will be less from the GI tract. Catharatics, which increases GI motility tend to reduce the absorption of any given medication. Cathartics, that means cathartics, they are the purgative. They are increasing the GI motility. They are used as a laxative and they are increasing the GI motility, and due to this it will reduce the absorption of any given medication. मतलब यहाँ पे क्या होगा, emptying time it will be raised in case of when the person is taking cathartics. Then propenthelin or atropine. Propenthelin, that is used to treat ulcer, and this is, we can say, it is a anticholinergic drug. And atropine, it helps to reduce the saliva or mucus and used to treat spasm in stomach, and it is one of the antidote in certain poisoning condition. So this propenthelin or atropine, it will decreases the GI motility and hence they will increase the absorption of drugs. देखिए यहाँ पे motility that will has been decreased by the particular drug. So यहाँ पे क्या होगा कि emptying time will be raised, and in this condition the absorption of particular drug, देखिए यहाँ पे one of the example is there, that is of the digoxin. So the absorption of the digoxin will be raised. देखिए बहुत बार क्या होगा कि अगर in certain condition if it is not required and even though the absorption of drug that has been raised, then there might be the chances of toxicity of particular drug. Then bacterial flora in alteration of GI tract, another one point that is the we need to consider, that is the bacterial flora of GI tract that plays very important role in the GI tract metabolism. Modifying or eliminating the intestinal flora with antimicrobials may alter the susceptibility of patient to a drug. For example, इसको understand करने के लिए हम यहाँ पे example लेते हैं. Bacterial flora in intestine, it contribute to the metabolism of digoxin. देखिए यहाँ पे digoxin जो drug है, that is object drug over here in this example. Then in this condition, bacterial flora, it plays important role to the metabolism of digoxin, and patient receiving tetracycline or erythromycin, this is precipitant drug, if they are concurrently administered, then prevent the inactivation of digoxin to increase their bioavailability. See, in this condition, क्या होगा कि bacterial flora in intestine, it contribute to the metabolism of digoxin, and patient receiving the tetracycline or erythromycin drug with this, and this concretely prevent the inactivation of digoxin. तो यहाँ पे क्या होगा कि inactivation, जो भी digoxin का यहाँ पे inactivation हो रहा था, that will be reduced or that will be avoided and that will be prevented. Due to that, the concentration of digoxin that will be more, and due to that the availability of the digoxin will be raised. So this is the effect of the bacterial flora because it plays very important role in the metabolism. Then alteration of content of GI tract. Now as a content we need to consider very first thing, that is the alteration of pH. देखिए pH है उसका भी बहुत बड़ा रोल होता है. pH अगर हम स्टमक के लिए कंसीडर करें तो generally the pH of stomach that will be on a acidic side. It will be nearly 1 to 3, in between 1 to 3. अगर हम intestine को कंसीडर करें तो intestine की pH है that is nearly 6 to 8. That will be near to basic side. So the pH that also plays very important role for the absorption, and also it may affect the drug interaction or it may induce the drug interaction. So here alteration of pH it may modify drug ionization, solubility and stability, and thereby markedly affect both the rate and extent of absorption. That means the absorption rate that is dependent on the ionization, solubility and stability. Most drugs are weak electrolyte which are most readily absorbed through the lipid containing membrane of the body, and when they are in the most lipid soluble form, and this is, we can say, that is non-ionized state. मतलब कोई भी drug non-ionized state में है तो maximum absorption होगा. Therefore, lowering the pH, if we are reducing the pH, increases the rate of absorption of weakly acidic drugs and vice versa. मतलब अगर यहाँ पे if you want to increase the rate of absorption of weak acidic drugs, then we should reduce the pH or we should decrease the pH. Then alkaline antacid in the stomach. If we are considering one of the example, alkaline antacid in the stomach, it inhibits the absorption of nalidixic acid, nitrofurantoin, oral anticoagulants, phenylbutazone, probenecid, and salicylates, etc. This agent that lower the pH tend to decrease the absorption of basic compound by making them highly ionized. Thus, the basic drugs, for example, aminopyrin, ephedrine, and quinine, they are not absorbed at the acidic pH of the stomach. मतलब यहाँ पे क्या है? Basic drug है aminopyrin, ephedrine, quinine, they are basic drug and they are not absorbed at the acidic pH of the stomach. मतलब ये सब ड्रग है वो एब्जॉर्ब नहीं होगी स्टमक में से. यहाँ पे स्टमक की जो भी pH है that is on a acidic side. Then alkaline antacid, here we have considered alkaline antacid in the stomach. It will inhibit the absorption of this acidic drugs, we can say. So this is regarding the alteration of the pH. Then complexation. The GI absorption of some drugs may be markedly decreased by the chelation with the aluminium, barium, calcium and magnesium. देखिए gastrointestinal absorption, if we are considering, if the product if any drug that forms complexation or we can say the chelation, then the absorption will be reduced or will be decreased. For example, tetracycline may not be absorbed in the presence of antacid and in presence of dairy product, because antacid and this dairy product, they contains metals in their product, so that's why they are not properly absorbed. And here there will be the formation of poorly soluble salt. If we are considering tetracycline, if we are taking tetracycline with the milk, then there will be the formation of poorly soluble salt, which is also known as a insoluble chelate, and it will not be absorbed in the body, and that will result in a therapeutic failure. That means tetracycline will not be absorbed in such a condition when it is taken in the presence of dairy product or it is taken in with the antacid due to the formation of the complexation. Then ciprofloxacin, it forms complex with divalent cation of antacid, leading to their decreased bioavailability. In such situation, milk, antacid or multivitamin, if we are taking, then the preparation containing such preparation, they are containing zinc and iron, should not be taken 8 hours before and 2 hours after the administration of the drug. तो यहाँ पे क्या है कि अगर हम कोई प्रोडक्ट ciprofloxacin अगर ले रहे हैं तो in such a condition, if we are taking the milk or if we want to take antacid, then in certain condition it should be taken before the 8 hours of taking drug of like ciprofloxacin, and after the और दूसरी कंडीशन ये भी है कि हम 2 hours के बाद अगर 2 hours के बाद भी ये प्रोडक्ट ले सकते हैं. So actually it should be avoided if there is a formation of the complexation, and due to that there might be the higher chances of drug interaction. So this is about the complexation. Then dissolution, deaggregation and dissolution rates of some drug that might be considerably decreased or accelerated by interaction. देखिए यहाँ पे दोनों पॉसिबिलिटी है, डिक्रीज भी हो सकता है और एक्सेलरेट भी हो सकता है. thus causing loss of efficacy on one hand, efficacy that means efficacy will be reduced in certain condition or increase the potency मतलब उसकी ज्यादा इफेक्ट आ सकती है, and in this condition there might be the chances of toxicity also. For example, an enteric coated drug like bisacodyl. Bisacodyl, that is also a purgative. So here the enteric coated drug like bisacodyl may be extremely irritating to the stomach if its coating is rapidly removed by the simultaneous administration of the gastric antacid. देखिए यहाँ पे क्या होगा कि bisacodyl है, bisacodyl that is a purgative drug and it is available in an enteric coated tablet. In normal case, the tablet it remain intact in a acidic fluid of stomach and it will be dissolved in a basic environment of the intestine. मतलब ये ड्रग क्या होगा कि स्टमक में वहां पे कोई इफेक्ट नहीं आएगा because there is a entry it is one type of coated tablet. तो coated tablet है तो ड्रग स्टमक में रिलीज नहीं होगा, वहां पे कोई इफेक्ट नहीं आएगी. So when it will have the favorable condition, that is the basic condition, in that condition the drug will start to release or it will dissolve in a basic environment. So in this condition, the product will give the proper effect. But if it is taken with the antacid or milk, which will reduce the acidity of the stomach fluid. Initially, we have taken the bisacodyl with the milk. In this condition, the acidity of the stomach that will be reduced. That means stomach के अंदर basic environment create हो जाएगा. Then in this condition, the tablet it will get dissolved and the drug that will be released in the stomach, and due to this there will be the severe gastric irritation and even the patient feel vomiting also. So dissolution that also affect the vital role regarding the drug interaction. Then diffusion, dispersion of a drug in the ambient fluid to its site of GI absorption may be inhibited if an interaction alters any one of the large number of physical properties such as miscibility and viscosity. So especially the product it will give the proper effect, that means the absorption will be proper, but in certain condition if the miscibility of product, miscibility of the drug and the viscosity of the particular environment, if it will be changed in certain condition due to the effect of some adjuvants, for example gelatin and carboxymethyl cellulose, then in such a condition the absorption will be inhibited, and this is also one type of drug interaction we can say. Then salt formation. Iron salt form poorly absorbed insoluble carbonates with antacid and other drugs that contain the carbonate radical. So in this condition this decreases the rate of absorption. तो यहाँ पे salt formation होने के कारण क्या होगा कि absorption that will be decreases. Absorption of iron salt it will be decreased. So these are regarding the GI tract absorption or alteration of the content of GI tract. Then alteration of the mucosa. If the intestinal mucosa is destroyed by toxic dose of agent like tannic acid that is used to stop the bleeding. So if the intestinal mucosa that is destroyed by the toxic dose of particular drug, for example tannic acid, the absorption of drug may be as rapid as intramuscular or subcutaneous injection. देखिए यहाँ पे क्या होगा कि intestinal mucosa अगर destroy हो रहा है तो destroy हो गया है तो in this condition the absorption rate will be increased, and this absorption rate intramuscular, अगर आप intramuscular और subcutaneous injection ले रहे हो तो इसके जैसा oral dose का absorption rate हो जाएगा. तो alteration of the mucosa it will most affecting the absorption rate. Then alteration of the transport mechanism. Drug interaction may interfere with an active mechanism by competing in the transport cycle. देखिए अगर यहाँ पे interaction that means active transport अगर हो रहा है तो कहीं पे अगर in this condition में it will also play the important role when there is absorption of the particular drug. For example, methyl dopa will be absorbed slowly from the intestinal tract in the presence of certain natural amino acid. That means this is competition of the transport mechanism. If there is a presence of amino acid, then it will solve competition, it will compete with the methyl dopa, and due to that there will be slow absorption of the methyl dopa from the intestinal tract in the presence of natural amino acid due to their transport mechanism. So this is regarding the transport mechanism, absorption. Then interaction during dermatomucosal absorption. Interaction during dermatomucosal absorption. A topically applied drug that is absorbed through the skin or membranes of the ear, eye, mouth, nose, rectum, urethra or vagina may interact with the drug when administered orally or parenterally. बहुत बार ऐसा भी हो सकता है यहाँ पे कि some topically applied drug, if you are giving some drug topically, then it may interfere with the drug that is administered orally or parenterally. Therefore, the drug interaction that includes the rats and site of dermatomucosal as well as GI absorption and function must be anticipated and considered. मतलब अगर हम कोई ओरल ड्रग दे रहे हैं और उसके साथ में कोई दूसरी कोई डर्मेटोम्यूकोसल एब्जॉर्प्शन के लिए भी हम कोई ड्रग दे रहे हैं, तो इन दिस कंडीशन वी नीड टू कंसीडर कि यहाँ पे एक दूसरी भी ड्रग दी जा रही है जो कि स्किन के ऊपर हम लगा रहे हैं, तो पर्टिकुलर लेयर के ऊपर हम लगा रहे हैं, मेम्ब्रेन के ऊपर लगा रहे हैं, तो यहाँ पे भी ड्रग इंटरेक्शन का चांसेस है. तो इसी कंडीशन में भी वी नीड टू कंसीडर अबाउट द dermatomucosal absorption of any other drugs. So when we are taking any drug topically, so these are the interaction during distribution. When certain drugs they are absorbed into the bloodstream, most of their molecules attach themselves to the plasma protein. The portion of the drug which is being transported in the bound form is pharmacologically inactive, and only the free molecule that diffuse into the tissue and exert their effect. मतलब यहाँ पे क्या है, the portion that is transported in the bound form में अगर प्लाज्मा प्रोटीन के साथ कोई प्रोडक्ट कोई ड्रग है, मॉलिक्यूल है, that is going to be bounded, then it will be act as a pharmacologically inactive. तो जो भी इफेक्ट हमें मिल रही है प्रोडक्ट की, कोई भी ड्रग है उसकी जो भी इफेक्ट मिल रही है, that is due to the free molecules. Now the presence of a second drug with a higher affinity for a protein, it will compete with the first drug for the protein binding sites and displaces the first drug, producing serious toxicity. देखिए दो ड्रग हमने अलग अलग दिए हैं, तो यहाँ पे क्या है, फर्स्ट ड्रग we have given, and in that condition it has been bound with the plasma protein. Then we are giving the second drug. Now this second drug, it has higher affinity for the protein, and in this condition what happened, it will displaces the first drug and it will be bounded. In this condition क्या होगा यहाँ पे कि पहली जो भी ड्रग है वो डिस्प्लेस हो गई है और दूसरी ड्रग है वहां पे ड्यू टू द हायर एफिनिटी इट हैज बीन बाउंडेड विद द प्रोटीन, प्लाज्मा प्रोटीन. तो यहाँ पे क्या होगा कि जो भी फर्स्ट ड्रग फ्री हो गई है यहाँ पे फर्स्ट ड्रग, it will give the higher absorption rate, and due to that there might be the higher chances of toxicity. For example, warfarin is 98% protein bound and only 2% is free in the plasma, and which is the active fraction. देखिए warfarin that is oral anticoagulant है, यहाँ पे only 2% that is free and which gives the active effect, or we can say active pharmacological effect. If another drug like phenytoin, it is given, competition for the binding site occur and warfarin is displaced. If the binding of warfarin is reduced to 96%, it would lead to doubling. That means 2% unbound to 4% is unbound. Previously देखिए only warfarin अगर दिया है तो there was 2% unbound, लेकिन यहाँ पे phenytoin भी अगर दिया गया then again it will give the competition for the binding site, and due to that 2% is more, it will be free, it will become 4% which is free and of the free concentration of the drug which will leading to the toxicity of the warfarin. मतलब यहाँ पे अगर अनबाउंड कंडीशन में ड्रग है तो यहाँ पे उसका एब्जॉर्प्शन ज्यादा होगा, मतलब यहाँ पे the chances of toxicity that will be raised. Another one example is there, that is tolbutamide. Tolbutamide, that is oral hypoglycemic medication, and that is used to treat type 2 diabetes. This tolbutamide can be displaced by dicumarol, that is also oral anticoagulant, resulting in a severe hypoglycemia. देखिए यहाँ पे क्या होगा कि अगर already we are giving tolbutamide and it has been displaced by the dicumarol, resulting in the severe hypoglycemia, that means कि यहाँ पे tolbutamide that will be in a free condition, free radicals we can free condition, तो यहाँ पे क्या होगा कि tolbutamide की इफेक्ट ज्यादा आएगी और इसके कारण we can say it will induce the severe hypoglycemia. Certain drugs which have a higher binding affinity to plasma protein displaces agent with a lower binding affinities. So in this condition also, the tolbutamide, it will give severe hypoglycemia. It will induce the hypoglycemia in this condition. So these are the interaction due to the during the distribution. Then interaction during biotransformation. The process of conversion of drug to their respective metabolite is termed as a biotransformation, and it usually occurs in the liver. Mainly the biotransformation, the process of biotransformation that will occur into the liver, and it is one type of, we can say, conversion of drug to their respective metabolites. मतलब biotransformation process से क्या होगा कि drug है that will be converted into the metabolites. This reaction are generally mediated by enzyme. देखिए ये पूरा जो बायोट्रांसफॉर्मेशन का प्रोसेस है इसको खत्म करने के लिए या फिर इनिशिएट करने के लिए it will enzyme, that is very much important, so that drugs capable of altering the enzymatic processes involved in the drug metabolism can lead to drug interaction. That means अगर कोई हमारे पास ऐसा ड्रग हमने लिया है जो which gives effect on enzymatic process or which gives directly effect on this enzyme which are very much required for the biotransformation process or metabolism process, then in such a condition there is a chances of drug interaction. The major hepatic enzyme system that consists the microsomal P450 or cytochrome P450. There are numerous forms of cytochrome P450 which is also known as a CYP. There is a very 20 to 200 different CYP, cytochrome P450 human genes, or we can say out of this CYP genes that have been identified, there are some different families which is known as a CYP1, CYP2, CYP3 and cytochrome P4. They are involved mainly, this family that is involved in the drug metabolism. So more than one isoenzyme that might be involved in the metabolism of the drug. For example, in the metabolism of propafenone, more than one isoenzyme they are involved. Furthermore, a drug may act as a competitive inhibitor of one isoenzyme while being metabolized by another. मतलब यहाँ पे क्या है कि drug it will act as an inhibitor also, and also it will be metabolized by one another enzyme. मतलब एक enzyme से inhibit हो रहा है. For example, quinidine, it inhibits the CYP2D6 isoenzyme but is metabolized by CYP3A4. मतलब एक enzyme को inhibit कर रहा है quinidine, it will inhibit one of the enzyme, and same time it will be metabolized by the CYP3A4. So this might be the effect in case of biotransformation. In this biotransformation, again we can divide them into the two portions, we can say enzyme induction and enzyme inhibition. So first we will consider about the enzyme induction. When a drug, we can say the inducer, increases the metabolism of another by stimulation of activity of drug metabolizing enzyme, then it is called as a enzyme induction. मतलब यहाँ पे initiation हो रहा है, यहाँ पे metabolism process के लिए initiation हो रहा है, increases the metabolism of another. One drug, it is increasing the metabolism of another by stimulation of activity of drug metabolizing enzyme. मतलब यहाँ पे enzyme है, उसको stimulation मिल रहा है. Then in such a condition, that is known as a enzyme induction. This result in a reduced therapeutic response of the drugs. देखिए यहाँ पे क्या होगा कि यहाँ पे stimulation हो रहा है, metabolize stimulation हो रहा है, तो drug है, drug that will be converted into inactive form. So due to that, the effect of that particular drug that will be reduced. This result in a reduced therapeutic response of the drugs that are metabolized by this enzyme. For example, enzyme induction का यहाँ पे example देखते हैं. For example, anti-epileptic drug such as phenobarbital, phenytoin, and carbamazepine are potent inducers of cytochrome, CYP3A4, other CYP isoenzyme, and enzyme involved in a phase 2 metabolism. This drug, they interact with the combined oral contraceptive, and due to that it reduces its effectiveness. देखिए यहाँ पे anti-epileptic drug है, anti-epileptic drug, they are inducer of particular enzyme, and this drug, if this anti-epileptic drug, they are induced, they are administered with the oral contraceptive drugs, then it will reduce the effect of contraceptive drugs, and also it will reduce the effectiveness of the contraceptive drugs. With low dose, देखिए यहाँ पे experiment के ऊपर से पता चला है, with low dose of oral contraceptive containing less than 35 microgram estrogen, pregnancies have been reported in patient taking enzyme inducing drug. देखिए यहाँ पे patient already if she is taking contraceptive medicine, and at the same time she was taking anti-epileptic drugs, then in this condition the pregnancies have been reported due to the enzyme inducing drugs. Enzyme inducing drug मतलब यहाँ पे हम anti-epileptic drug है, they are the enzyme inducing drug. तो यहाँ पे क्या होता है कि क्या हुआ कि enzyme induction यहाँ पे हुआ है साइटोक्रोम CYP3A4 का, तो इसके कारण यहाँ पे ओरल कॉन्ट्रासेप्टिव की जो इफेक्ट होनी चाहिए या फिर जो भी इफेक्ट उसकी होनी चाहिए, that has been not initiated, and due to that the effectiveness of this contraceptive has been reduced. So this is the one of the example of the enzyme induction. Then another one, that is the enzyme inhibition. When one drug, the inhibitor, we can say, decreases the metabolism of another. It decreases the metabolism of another by inhibiting or inactivating the drug metabolizing enzyme. It is called enzyme inhibition. यहाँ पे एक drug है that will inhibiting or inactivating the drug metabolizing enzyme, so that enzyme will be initiated in, और यहाँ पे ज्यादा drug मिलेगी अगर inactivation हो रहा है या फिर कम हो रहा है तो इसके कारण यहाँ पे free drug मिलेगी. This result in a potentiation or therapeutic response or precipitate toxicity of the drugs that are metabolized by this enzyme due to the accumulation of unmetabolized drugs. देखिए यहाँ पे क्या होगा कि there will be the accumulation of unmetabolized drug. Unmetabolized drug है उसका concentration यहाँ पे बढ़ जाएगा, तो जो भी ड्रग का इफेक्ट है वो यहाँ पे ज्यादा है. देखिए previously in case of enzyme induction में क्या था कि वहां पे कम इफेक्ट आ रही थी, वहां पे मेटाबोलाइज प्रोडक्ट हो रही है तो वहां पे इफेक्ट कम आ रही है. लेकिन यहाँ पे क्या होगा कि अनमेटाबोलाइज ड्रग ज्यादा है, अनमेटाबोलाइज ड्रग ज्यादा है, तो इसके कारण क्या होगा कि थेराप्यूटिक रिस्पॉन्स ज्यादा आएगा. थेराप्यूटिक रिस्पॉन्स ज्यादा आएगा तो देयर माइट बी द चांसेस ऑफ टॉक्सिसिटी इन दिस कंडीशन. We will take one of the example. For example, the anticonvulsant phenytoin is metabolized by the hepatic microsomal enzyme, and its effects can be potentiated by microsomal enzyme inhibitor like para-aminosalicylic acid, disulfiram isoniazide, and methylphenidate due to the accumulation of unmetabolized drug. मतलब यहाँ पे क्या होगा, phenytoin. This increased level of phenytoin then that can result in a toxicity. Phenytoin is metabolized by the hepatic microsomal enzyme, and its effect can be potentiated by microsomal enzyme inhibitor like para-aminosalicylic acid, disulfiram isoniazide. मतलब यहाँ पे phenytoin और isoniazide दोनों साथ में दी गई है अगर ड्रग, then there will be the accumulation of unmetabolized drug of phenytoin. So in this condition, the phenytoin concentration that will be raised, and due to that there will be the chances of toxicity. So this is one of the example of enzyme inhibition. Then interaction during excretion. Interaction occurring during the excretion of drug may involve any one of the renal excretory process. It means glomerular filtration, tubular reabsorption, or active tubular secretion. Most important drug interactions that occur as a result of either change in a urinary pH, which alters the function of the reabsorbed drug, or through the competition for active tubular mechanism. देखिए यहाँ पे excretion में अगर हम कंसीडर करें कोई ड्रग इंट्रैक्शन हो रहा तो मेनली देयर माइट बी ड्यू टू द changes in the urinary pH. Urinary pH का pH अगर change हो रही है, then in that condition there is a chances of interaction, and another one, that is the competition for the active tubular mechanism. So we will see this changes in urinary pH. The urine pH, it determines the extent of ionization of the drug in the renal tubule and affects the fraction of reabsorbed drug. Acidic drug remain in unionized form in acidic urine, whereas alkaline drug remain unionized in alkaline urine. Now when drug is in unionized form in urine, it diffuses easily back into the blood from urine and shows prolonged or intensified action. देखिए यहाँ पे अगर हमें कोई प्रोडक्ट है, कोई ड्रग है, उसके ज्यादा इफेक्ट चाहिए, then in that condition, if the drug that is in unionized form from in a urine, अगर यूरिन के अंदर unionized form में है, then again it will diffuse back into the bloodstream and it will shows the effect of particular drug. But अगर यहाँ पे ionization अगर हो गया, then it will be excreted. See यहाँ पे acidic drugs remain in unionized form in acidic urine. Acidic drug, it will remain as it is, or we can say it will remain unionized in acidic urine, whereas alkaline drugs, it will remain unionized in alkaline urine. Now acidification of the urine with agent like ammonium chloride result in a reduced effectiveness of a basic drug, for example amphetamine or quinidine, as they will be largely ionized in acidic pH and readily excreted. देखिए यहाँ पे basic drug है, that will be excreted if it will come in a acidic pH. In acidic pH, it will be converted into the ionized form, and due to that it will be readily excreted. So यहाँ पे अगर हम acidification process करें तो यहाँ पे it will be helpful for the excretion of the basic drugs. In contrast, renal excretion of acidic drug, if you want to excrete the acidic drug, for example salicylate, barbiturates, anticoagulants, it will be accelerated by the alkalinization of the urine, and we can initiate the alkalinization of the urine with the sodium bicarbonate, acetazolamide, or potassium citrate. Thus ammonium chloride may be given in a amphetamine overdose. If there is an amphetamine overdose, then we can give the ammonium chloride to initiate excretion, to increase the excretion of amphetamine. So in this condition of the overdose of amphetamine, we can use the ammonium chloride, and whereas sodium bicarbonate, that may be used in phenobarbital or salicylate, that means acidic drug overdose. अगर एसिडिक ड्रग सैलिसिलेट या फिर फिनोबार्बिटोन का ओवरडोज है, then in this condition we can give the sodium bicarbonate and which can initiate or which can increase the excretion of the acidic drug. So these are the main effect due to the changes in the pH. Then competition for tubular mechanism. See, many drugs and metabolites, they are actively secreted from the renal blood vessel into the tubules and subsequently they are eliminated. मतलब कोई drug या metabolite है, they are actively secreted in renal blood vessel and then afterwards it will be eliminated. When true drugs utilizing, they are having the same transport mechanism, are present at a time in a blood or tubular urine. If we are giving the two products at the same time and they are having the same transport mechanism, in this condition, these two drugs, they will interfere in the excretion process of each other during active tubular secretion process, and due to that, it will initiate the interaction in between the drug. For example, verapamil, it inhibits the renal tubular secretion of digoxin when concurrently administered and increases the serum concentration of digoxin, and in this condition, due to the effect of verapamil, it will inhibit the tubular secretion of digoxin, and in this condition, there might be the chances of digoxin toxicity. Then competition for active tubular secretion can be used therapeutically as well. मतलब बहुत से केस में ये अच्छा भी है. अगर हमें कोई ड्रग की ज्यादा इफेक्ट चाहिए, then we can use such a type of combination. For example, probenecid with the penicillin to delay the rapid excretion of the penicillin molecule, leading to a significantly prolonged effective duration of action of penicillin. देखिए यहाँ पे if we have given probenecid with the penicillin, then this probenecid, it will delay the excretion of the penicillin and it will compete for the transport mechanism, and due to that the penicillin, it will remain in the blood, and due to that the higher effect, or we can say the prolonged effect of the penicillin, that will be observed. So this is the good effect, or we can say this is the good combination for a higher effect of particular drug. So we can use this tubular mechanism. This is one of the good side. Then, so these are regarding the pharmacokinetic drug interaction. We have considered about the absorption, distribution, metabolism and excretion over here. Then second one, that one is a pharmacodynamic drug interaction. Drugs, they are having similar pharmacological effect. A patient, they are receiving hypnotic, may develop an allergic skin reaction, and for this he receives an antihistamine. In last lecture we have discussed about this, which itself has a central nervous system depressed effect. मतलब अगर कोई patient है, already he is taking hypnotic, and if he develops, he is having the skin reaction. In skin reaction, if doctor prescribe about the antihistaminic, and this antihistamine, they are also having central nervous system depress effect. तो यहाँ पे क्या होगा कि synergistic effect यहाँ पे मिलेगा. दोनों ड्रग है, दोनों ड्रग की एक जैसी इफेक्ट है. So the combined effect may be sufficient to produce serious sedation, which cannot be obtained from the either of these drugs. अगर ये ड्रग हमने इंडिविजुअली लिया है तो वहां पे कोई सीरियस सीडेशन जैसे इफेक्ट नहीं आएगी, लेकिन अगर दोनों ये ड्रग कॉम्बिनेशन में लिया है तो in this condition there will be the serious sedation. Then similar effect they are also observed with the use of hypnotic and alcohol. This is also CNS depressant, both having the CNS depressant effect. The narcotic analgesic and tranquilizer increase the CNS depression, and digoxin and quinidine, it will induce the bradycardia. यहाँ पे bradycardia, that means it will show the slow heart rate, or heart action will be reduces. So these are the similar pharmacological effect that is in a pharmacodynamic drug interaction. Then drugs having opposite pharmacological effect. When a patient is receiving pilocarpine, that means cholinergic drug, for a glaucoma, that is for a eye disease, and at the same time for abdominal pain may be prescribed with an anticholinergic drug. Anticholinergic drug also prescribed, and if the patient is taking with the pilocarpine, then intraocular pressure may be altered by the anticholinergic drugs. So the effect will be nullified. The same example we can say if the patient is taking hypnotic drugs and same time he is taking caffeine. So the hypnosis that means the sedation, that will be opposed over here. Though this one is a opposite pharmacological effect, that is also known as a antagonistic effect. Then change in a electrolytic and fluid balance. See, if the drugs, for example diuretic, that cause the potassium depletion may potentiate the effect of digitalis and non-polarizing muscle relaxation, but it will antagonize the effect of lignocaine, quinidine and procainamide. मतलब यहाँ पे diuretics सब one drug, it will cause one of the effect towards the it will potentiate the effect of digitalis के लिए ये potentiate effect देगा अगर diuretics that is taken to the patient, that is given to the patient, and same time it will show the antagonistic effect of towards the lignocaine and quinidine and procainamide. So this is due to the change in the electrolytic and fluid balance. So these are regarding the pharmacodynamic drug interaction. So here in this video we discuss about the pharmacokinetic interaction. In that we have considered the drug interaction during the absorption, drug interaction during the distribution and plasma protein binding, interaction during the biotransformation, and interaction during the excretion. And also we will discuss about the pharmacodynamic interaction. Thank you for watching this video.